Okay, welcome. Today, I will be talking about SDF-Bayes. This is our latest work in safe dose finding clinical trial methodology design that specifically deals with drug combinations and the heterogeneous patient groups. My name is Shen. I'm assistant professor at the University of Virginia. This is a joint work with my collaborators, Hyun-Sok Lee, Bill Zame, Jian-Wang Lee, and Mihaela van der Schaar. This talk is based on our upcoming AISTATS 2021 paper, which I invite you all to check out. Let me start with a little bit of a background. Dose finding clinical trial is also known as phase 1 clinical trial. The purpose of this trial is to find out the so-called MTD, maximum tolerated dose of a new drug or certain drug combinations. In this work, we focus on drug combinations because they are more useful for certain diseases in practice. And the objective here is to find the dose combination DC, whose overall toxicity is closest to our target. And this DC is called the MTD. The main challenge associated with this task is how can we do this safely? Because phase 1 clinical trial is acting on human participants. So we want to be very careful about exposing these participants to unnecessary overdose. The safety constraint we enforced in this work is the average toxicity of trial to not exceed a target threshold. Let me explain this trial using a simple example of just one drug with 4 dose levels. Here, we do not know the toxicity level of each individual dose. Our goal is to find out which one of these 4 doses has a true toxicity level that is closest to our target, let's say 0.35. If we know the underlying ground truths like this, then we know that dose level number 3 is our MTD because 0.3 is the closest to the threshold of 0.35 among all these 4 dose levels. And the problem obviously is how can we learn this through the trial? And the additional challenge, as I already mentioned, is the safety of participants. In this example, dose level number 4, 0.4 milligram, is considered an overdose. And the toxicity level is higher than our threshold. And we do not want to expose many participants to this level. So we want to minimize this exposure. The main challenge in clinical trial with drug combination is mainly coming from this fundamental conflict between the exponentially increasing DC candidates and the limited patient budget. On the one hand, we have the number of DC candidates that would increase exponentially with the number of drugs. On the other hand, we only have a limited number of patients. And in reality, we are dealing with maybe a dozen, several dozens, or maybe to 100 patients. However, the number of DC candidates can grow quite large. So how can we effectively learn the MTD with limited patient budget? This is really the key problem. Luckily, we have very good parametric dose toxicity models from the literature and from practice. And the idea here is for us to, instead of learning this toxicity level for every DC candidate, we're going to reduce that to learning the parameter of a particular parametric dose toxicity model, and then using this parameter to estimate toxicity probability of all the DCs. This would help us to reduce the requirements in terms of having limited patient budgets, while we still want to learn the MTD effectively for a large number of DC candidates. So let me get to the details of SDF-Bayes. As the name suggests, this is the Bayesian design. So we learn the parameters via a Bayesian approach. On the other hand, this is an adaptive design, meaning that we are making decisions on the fly based on the past observations. More specifically, we choose the DC to be tested next based on the current estimation of the dose toxicity model, is in particular with the learned parameters. And once we get this estimation, we're going to make the decision which implicitly uses past observations. The operation goes like this. So in the current round, let's say I have a decision already that has already been made. We're gonna test the DC of 0.1 milligram and 0.3 milligram. And we're then we will give this DC to the trial participants, and then we observe the toxicity outcomes. Based on this outcome, we're gonna update the parameter theta of the dose toxicity model. With this updated model, we're gonna make the next decision. So what is the next DC we're going to test? And we're going to continue this loop for a given number of rounds, which is determined by our patient budget. So more details about SDF-Bayes. How does SDF-Bayes really work? This algorithm actually runs on two fundamental principles. The first principle is optimism. This is really for efficiency. In a nutshell, we want to choose the most likely DC to be the MTD as a DC to be tested next. So we want to be very aggressive in terms of selecting the DC that looks the most promising in terms of being the MTD. And this is done by solving this optimization problem based on the current estimation of theta. And we are going to have a toxicity probability, which is a posterior probability that is constructed for every DC. And for this DC, we're going to, let's say A, we're going to try to evaluate the current estimation to the threshold psi. And we will look at this target interval and pick the DCA that has the largest, actually, sorry, the smallest interval that is around psi. That's going to give us the choice of the next DC to be tested. So in this particular example, after doing this evaluation, we realize that the probability of this particular combination, this DC, is the largest based on the current estimation of theta and based on the threshold psi and the u. And we're going to try to test this DC next. So this is purely based on optimism. Again, we are trying to improve the efficiency. We want to be very efficient in terms of getting to the MTD as quickly as possible. The other side of the coin, though, is the safety, which brings up the second principle. This is the cautiousness. We want to be conservative to incorporate the safety constraint. The idea here is that we want to constrain the choice of DC to only a subset of DCs, where for this subset, based on the posterior distribution of each DC, we evaluate this subset to be unlikely to violate the safety constraint that we have, given before, like this. This is our safety constraint. So we want to be conservative based on our current best knowledge, and then we want to, control our action within this set. So in this particular example, we have this kind of, conservative expected toxicity, and then if the result is such that our previous choice is unlikely to violate safety constraint, we're going to choose this choice. If, on the other hand, we have a situation where this choice actually falls into the likely to violate safety category, we're going to actually discard this choice and choose the next best option. This is the overall SDF-Bayes operation. Okay. So next, let me move to the heterogeneous patient group situation. In this case, we are dealing with heterogeneous patient groups where instead of having one population for all, we are having subgroups. For example, we divide the population to male and female, or based on the age of the patients, we have different subgroups, and so on and so forth. Mathematically, we have M pre-identified heterogeneous groups, and we have a total budget of T patients. Now, instead of learning the MTD for the entire population, the goal becomes even more ambitious and more difficult. We want to find the MTD of each subgroup. So the idea is, how can we allocate this T patients adaptively to improve our learning across this, let's say, 3 different subgroups? The idea for SDF-Bayes AR is to do adaptive recruitment for patients. The mathematical tool here is that we're going to estimate an expected improvement for every subgroup, and then we choose the subgroup that has the largest EI. Then we're going to use this group for the next round. And this expected improvement is computed based upon the additional patients. Adding one more patient, what is the benefit? And we choose the subgroup with the largest benefit. So let me actually talk about experimental results. In this example, we have 3 synthetic data set and a real world data set. And we compare, first of all, we compare SDF-Bayes to the baseline methods for the homogeneous group situation. And our results actually demonstrate that SDF-Bayes satisfy the safety constraints while making fewer errors than the state-of-the-art solutions. There is one exception, though. Here, the DF-Bayes actually has a better recommendation error than our method for synthetic data set B. However, This comes at the cost of unacceptable proportion of safety violations, as you can see here. Lastly, the heterogeneous group situation. We also see that adaptive recruitment really improves SDF-Bayes and SOTA-Bayes. I should also emphasize here that the AR principle is not limited to SDF-Bayes. It can be applied to also other methods as well. This improvement, I want to finally make a comment here, is more significant when you have prior information that you can leverage to construct this prior distribution. And this is practically often possible because before the phase 1 clinical trial, you already have the drug development phase. You also have some animal trial. So there are useful data that can be leveraged to construct such prior information. That concludes the talk for today, and I'll be happy to take questions. And thank you for watching.